Not the real website. Okay. Okay. Thanks all, and apologies for the inconvenience. I'm doing one last thing. I'm just trying to turn the waiting room off so people can come straight in. Okay. Okay, we're good to get underway. Apologies for the delays. So I should start by saying, well, firstly, thank you for having me here and to give this SEEC Toolbox seminar. This is not really going to be a stats seminar. It's going to be a predominantly an R tutorial, and there will be a little bit of stats in it. And if you are not able to sit through the whole thing, or you find my dulcet tones too soporific and you decide you need to go have a nap, you can find the tutorial on the Say on Fanboss Nodes webpage, and I'll show you the link in a second. So you don't have to sit through all of this if you have better things to do, but it'll probably help you have some idea of what's in the tutorial if you hear me out, and then next time you can use it to find just the things you need. So we've recently launched a revised Say on Fanboss Node website. I just thought I'd show it to you if you're keen to have a look and explore. It's got the usual about us, about our science, a bit about our team, and you can click on the things and find out about the different people and the students. Even Craig's on there if you want to find out more about the mystery man behind the Toolbox seminars. There's some dashboards looking at stream flow and rainfall and the like, a bit about our research and study sites, some about our current projects, and then kind of news, publications, how to find us, that kind of thing. But up here, you'll see a tab called Data Skills, and what we're trying to do is kind of develop a set of kind of walkthrough tutorials using different kinds of data sets. So both teaching kind of data analysis, but also kind of presenting some of Seon's data sets for you to fiddle with and explore if you're keen. I'm just trying to find Craig. If anything pops up in the chat window, will you let me know? Because I don't seem to be able to find it.
Yeah, will do.
Cool, thanks. So on this page, we've got various data skills. There's a kind of a basic introduction to R. There's a kind of a motivation piece about why you should learn R, a basics introduction, and then the topic of today, which is analyzing biotic community data. Give it a second. And so I'm just going to walk you through this tutorial that I put together. It is pretty much an exercise in data wrangling to allow you to use the various functions in R to analyze your data. So there's a million different ways of analyzing biotic community data. There's a million ways of doing it just within R. If you check out the Envirometrics Task View, you'll find, I think it's over 100 libraries, each with 20 plus functions, some with over 100 functions. So covering this topic in a 1 hour session is just not possible. But the one major issue with all these packages and functions is that they each have their own kind of specific format requirements. So if you want to use a, if you want to do a generalized joint attribute model with GJAM, they have specific input requirements, and you have to wrangle your data to get it ready to plug it in. So kind of the point of this session today is more to show you some of the tips and tricks for wrangling your data and getting them into the right shape. But then also highlighting some of the issues in analyzing different kinds of biodiversity data as you go along. So there's a little bit of theory, but that'll be very light. Okay, so essentially, I only use 3 packages, although I don't think I even used vegan in the end. But vegan is a useful one to be aware of because it has a lot of tools for vegetation analysis. It follows the kind of base R syntax and functions. So it's not a tidyverse package by any means. but it has so many cool tools that it's unlikely to go away in a hurry. So I set the working directory for where the data set is, and you'll want to change this if you're actually trying to run this code. And then the data set you can actually download here. So it's from a paper published in 2017, and it's a vegetation survey or a repeat vegetation survey from the Cape of Good Hope section of Table Mountain National Park. In effect, it's 81 vegetation plots across the park. Each plot is 5 by 10 meters, so 50 square meters, and then surveyed 3 times in 1966, 1996, and 2010. And if you want to find out more, there's a metadata tab in the spreadsheet, and there's also the paper that you can read. So let's have a quick look at the workbook. If we use this function Excel sheets, it just tells us what worksheets are in the Excel file. And in this case, there's 11 sheets, the metadata one I mentioned, and a bunch of others. But we're only interested in a few of these at this stage. So usually when analyzing biotic community data, you usually have 3 separate data objects. And these are usually a species list or a species by site matrix, or often called the community data matrix, where you have, if it's in matrix format, you'll have species on usually as the columns and the sites as the rows, though sometimes they want it the other way around. And then the values in it are either presence absence, counts of individuals, or relative abundance, percent cover, biomass, that kind of thing. So that's the kind of like the primary data set that one would be analyzing, the species by site matrix. Then you usually have an environment matrix, which is usually measures of different environmental variables by site. So things like, if you're dealing with plants, it'll be information about soils, climate, location, and that kind of thing, water availability, and so on. And then often there's a trait matrix, so traits by species. So it'll be your list of species, and then what growth form they are, or what other functional traits they have. So if we look at our sheets, we've got we can see we've got environment. I spelled that wrong when I submitted the data. Oops. But essentially, we've got our environment tab, and we've got a traits tab. And then for the species by sites, we've actually got 3. We've got veg 1966, veg 1996, and veg 2010. And that's because this is a repeat vegetation survey across those 44 years. So in this case, we've got 3 of the species by site matrix. Okay, so we can have a quick look at them. Just to glimpse the top of the file, we can use the read Excel function to kind of give us the top left corner. So here I've said, give me A1 to K5, so it kind of draws a little corner. And here we can see, okay, we've got species as columns, and we've got plots as rows. So this is a community data matrix as we were expecting to find. I'm not going to describe too much about the functions as we go. There'll be a few that I'll highlight, but others you can work out for yourself. Read Excel is nice because it reads both XLS and XLSX, and it works it out for you. It's a little bit slow, but it's usually not an issue. Okay, so to look at the data, it's this community data matrix. There's lots of zeros because there's locations where the species didn't occur. So Acacia saligna wasn't in plot 1, which was a relief because it's an alien plant. But over here you can see at Adenandra villosa, there were 3 individuals in CP1. In actual fact, I don't know that there were necessarily 3 individuals because there's a flaw in this data set that I should highlight now before you think, oh, this is great, and I'm going to go write a paper using these data, which you are welcome to do. I mean, the data is free and open access, but you should be aware of the blemishes. And this blemish is essentially that the surveys done in 1966 and 1996, the field botanists didn't actually count all individuals of each species. What they did is they estimated abundance classes. So if there were between 1 and 5 individuals, it was given a class 1. If they were between 5 and 20, then it was given class 2, 20 and 50, class 3, and so on and so forth. And what I've done here is I've converted back to an estimate of the actual abundances by simply assigning the midpoint of each of those classes. And that's the case for both the 1966 and the 1996 data sets. And so this isn't necessarily defensible. It's up to you to justify to your reviewers and editors and yourself. So be that on your conscience. The 2010 data set is actually actual counts of individuals. So then we read them in. So read them in, have a quick look. Okay, it's species by sites, values inside. Here's a long list of the species that are in the site. I think altogether in the survey there were almost 600 species. And here's, so at the moment we're just going to look at the 1966 and the 2010 survey. So the first and the last surveys. We're going to ignore the middle survey. Before I move on though and start analyzing the data, just something to cover is that you don't always get your data set in a species by site matrix. So this is, you know, the community data matrix or the wide format. But often biodiversity data sets are stored in long or list format. So you usually have 3 columns, sometimes more. But usually if it's 3 columns, there'll be like the site code, the species name, and the abundance or the like. And it'll then be a much longer data set rather than this, this matrix with species on one axis and sites on another. So you need to be aware of how to convert between this kind of long format, which is also kind of more in line with the tidy data format, for those of you who are familiar with the tidyverse set of R packages and philosophies. So it's more in line with that format, but it's useful to be able to convert between the two, especially because if you want to use the tidyverse packages, you usually have to have the data in tidy format. If you want to use other packages like vegan and so on, then you actually need it in the matrix format. So in this case, we've read our data in a matrix format. So let's first look at how to convert it into long format. And there's this incredibly useful, well, there's 2 incredibly useful functions in dplyr and the dplyr package called pivot longer and pivot wider. And they essentially are for converting between kind of long and wide format data sets. And so in this case, we've said, take our data set, which is called species 66. Craig, I don't know if that's you or if there's somebody else who's not muted. I'm getting funny sounds like you're playing a gambling game.
Hi guys. Sorry, Jasper. Hi guys. Do you guys just mind muting your mics?
Thanks. Cool. So there's these 2 functions, pivot longer and pivot wider. And what I'm saying here is essentially take my wide matrix of the data from 1966 and apply this function. I say drop column 1. So cols give me all the columns apart from the first one, -1. And that's because if you look up here, the first column is actually the site names or the row names. And this is something I haven't got used to in the kind of tidyverse way of dealing with things, but they don't like having row names. And so your site labels have to be a column in the data set. And so when you analyze it, you always have to tell the functions to leave it out. So in this case, I say, okay, take that matrix, leave out column 1, although it will use it, as you'll see in a second. I want to send all the column names, which is the species names, to a new column called species. I want to take all the values in the matrix and put them into a new column called abundance. So it's pretty straightforward. You say, yeah, take the columns, take the column names, make them a new column, take all the values and make them a new column, and don't do that to my plot labels. And what happens is any of the columns that are excluded here actually become their own column in your long format data. So in this case, we said told it to leave that out of this kind of conversion, and it did, but it made it a new column in the beginning, which is what we want, because we want to know what site our data is in. So hang on. So here we have essentially site, species, and abundance. You'll see that my data set has a funny label for that first column, and that might be different depending on what operating system you're on. There is a note in here that talks about it. So if you're trying to run this code and you're getting errors, you might just check what that name is and either make it this or adjust it when I use that column name elsewhere. Okay, so that's pivot longer. There's also all kinds of other cool things you can do with pivot longer, and I think it's a function you can spend quite a lot of time getting to know because it's incredibly useful, but some of the ways of using it can be a bit complex and counterintuitive initially. Okay, so now we've gone from wide format data to long format data, and now we want to be able to do the inverse, and as I said, that's another function called pivot wider. And in this case, it's a similar syntax, but just the inverse. So we've said, okay, the ID column is column 1, which was our sites, or what would usually be thought of as your row names. And we'd say, take the column names from the column labeled species, and take the values in the matrix from the column labeled abundance. And then what I've added here, which is unnecessary in this case, but it would usually be necessary, is I say values fill equals 0. And the reason is, as you can see by this matrix, there's a lot of absences, 0. So Adenandra uniflora didn't occur in site 1 and it got a 0. But if you were storing your data in long format, usually these absences are implicit. You leave them out. So it's a big advantage of having the long format data is you only need to record where your species did occur. So looking at this, your entries in the long format data from this would be CP1 Adenandra villosa 7, and then you'd skip all these others, and then you'd go CP, I think this is 11 Adenandra villosa 7. And then the next one you get would be all the way down here, CP15, so you don't have to worry about all the 0 data. In this case, when I did pivot longer, I didn't remove the 0s, so my data set actually does have them in, but it's unnecessary. I didn't need them. So just a reminder that if you are going from long format data to wide data, you need to add some kind of value that fills the gaps. It can be NA, it can be anything you like, but usually it'll be 0 because it indicates the species is absent. Okay, so and I explain all that here, and I show that the two objects, the original object and the pivoted object, are the same. So now let's start looking at analyzing the community data. There's a very nice base R function called row sums, which is quite convenient for this. So now just a reminder that we're back to the wide format data, the community data matrix with species as columns and sites as rows. And so if you say row sums, that essentially sums all the values in each row or in each site. And so that would give you the number of individuals in each plot, just applying this function. Of course, this never works the first time, as anyone who works with R knows, and you get a completely useless error message like this. It says error, blah, 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 X must be numeric. And in this case, what that means is that one of our columns in this data set is not numeric. And we know that that's the first column, that's the plot column. So of course, I just need to rerun this and exclude that first column, which is what I've done here. And there's multiple ways of doing this. This is just to demonstrate that there are a million ways of doing anything in R. So here I've just selected column 2 to the number of columns. So that's everything but column 1. Another approach to do exactly the same thing is to say, just drop column 1, -1, drop 1. Or if you want to get all fancy and start using the tidyverse approach, things get a bit more complicated. Those of you familiar with the tidyverse will be aware that it can be incredibly powerful, but getting your head around the new and different ways of coding can be quite painful. And so I find row sums, everything but column 1, to be quite intuitive, whereas this, where I've said the data matrix without column 1 apply row-wise a mapping function, which is the sum. It seems like a over-elaborate way of getting to the same solution. Of course, this is much faster, the tidyverse approach, and you could then also add extra, you could pipe on extra functions if you wanted to go further down the line. So it is valuable to explore that way of doing things. Either way, this isn't very useful. This is just a long string of numbers, and it's quite hard to kind of make any sense of it. So let's plot it as a histogram, and this is what we get. So this is the count of numbers of individuals across the plots. And in this case, it goes from, we have some plots with 50 individuals, and we have some plots with almost 2,500 individuals. So there's a massive variation, and these are all plots that are the same size. It's all 50 square meter plots, and it's all essentially fane-boss vegetation or allied fane-boss vegetation, things like that. There's a few coastal dune strandveld or coastal dune thicket plots in here as well, and a couple of kind of more true wetland as well. So that's a huge variation, but this will become apparent why in a minute. So now, okay, that's abundance. Now let's see about calculating species, measures of species diversity. So the first one we're usually interested in is the species number, or the count of species. And in this case, you can do the same row sums thing very easily, but you just add this logical statement, and it'll give you species numbers. So this logical statement just says, is the value greater than 0? Which in this case would turn this matrix from 0s, 7s, 30s, and all the different numbers to trues and falses. And R treats a true as a 1 and a false as a 0. So this would be false, true, false, 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 blah, blah, true, and so on. But those are factors, and false is 0 and true is 1. So you can just say the sum of that column of trues and falses will give you the counts of numbers of species. So that's what I've done here, just by adding that little bit, and that gives you all of these numbers. And if we histogram them, we get this, which is essentially we have between 5 and 80 species across this set of plots. What about the relationship between the two? Okay, so now we've got, so here I've taken, I created a data frame object there, and then I plotted as a histogram, not a histogram, sorry, as a XY scatter graph. And so here create a data frame with 2 columns. One's called species number, and the other one's called abundance, and then plot it as an XY scatter plot. And then I've added a logistic curve just to kind of approximate the shape of the relationship. And as you can see here, the more individuals there are, the more species there are, but to a point. And this is the classic kind of species accumulation curve that one would usually expect to encounter. The shape of this relationship is not necessarily logarithmic, and this is the topic of much research and many, many, many papers, and everyone has their own favorite function that they try to fit from the zero-sum multinomial by neutral theory and the maximum entropy theory of species abundance distributions and all those kinds of things. So I'm not going to go there, but just to make you aware that this relationship between species number and abundance is something that's quite fundamental in ecology, and there's a lot of research focused on this, especially because the idea of being able to sample a small area or a small number of individuals and trying to project how much diversity you might have in the landscape. So the more diverse the landscape is, the steeper you would expect this curve to be. So in this case, this is actually quite a steep curve. If you were to be sampling forests or some other less diverse system in our area, you would have a curve down here that kind of saturates much more quickly. So the slower it saturates, the more diverse it is. Okay, but this also represents a problem for us. So in our data set, this these are all plots that we want to compare to each other, and we have this kind of limit on the numbers of species we get because of the numbers of individuals. So in effect, here in this plot, we've got probably 50 individuals, and you're not going to get more than 50 species in a plot with 50 individuals because it's just not possible. Whereas if you have 2,500 individuals, hypothetically you could have 2,500 species if this was an incredibly diverse system, more than the incredibly diverse system it already is. So this is a constraint, and this is something one needs to be aware of when dealing with sedentary organisms. So typically, in most kind of fields of biology, you calculate your species richness based on the numbers of individuals you've sampled. So this is common in zoology, things like trawl netting or mist netting or aerial counts and that kind of thing. Your species richness is some function of the numbers of individuals that you've collected. This is why I've been talking about species number so far, not species richness, because what I've been presenting isn't actually species richness by kind of some of the technical definitions. When working with sedentary organisms like plants, you usually sample per unit area. And that means you're sampling something completely different. You're actually sampling species density. So what is the density of species per unit area is what you're getting, and not species richness. This is not the end of the world. Species density is actually an incredibly useful metric to be able to measure. I'm just saying that it's not true species richness. And it can be biased in many different ways. So the classic example in fynbos is that fynbos burns. So a few years after fire, you're going to get 2,500 individuals and lots of species in a small plot of 5 by 10 meters or the like. But after the vegetation is 30 years old, maybe you're only going to sample a couple of hundred individuals. And so your species densities are going to have gone down in a big way. And previously, people have used this as an argument that, your fynbos needs to be burnt to maintain its diversity. But that's just simply not true. It's just a function of the age the vegetation was at when you sampled it. So it's just something to bear in mind. And that's a big problem when analyzing vegetation data sets, especially in a fire-prone system. It's also an issue if you were comparing fynbos to forest, for example, because obviously in forest you get much bigger individuals, so you're going to have fewer species, and it really just isn't comparable. You shouldn't be talking about richness. You can be talking about species density. I often find it's actually very useful to contrast species density and species richness, and so I'll come to how to calculate species richness in a minute. I haven't gone into this in much detail and in a particularly intelligible way, but I would highly recommend that you read this paper by Nicholas Gatelli and Robert Colwell. It's probably one of the highest cited papers in ecology, or in community ecology anyway, and it really, really is, it's earned that accolade. It's a worthwhile read to help you understand the kind of the funny things numbers do when trying to sample diversity, and the pitfalls you should be aware of. Okay, enough of that. Okay, so now to compare between surveys, what I've done here is I'm doing trying to draw a XY scatter plot comparing the species counts from 1966 with the species counts from 2010. And I get an error there. And essentially, that's because in our data sets, there's different numbers of rows and columns. So in 1966, they sampled 81 plots, but in 2010, we were only able to find, I think it was 63 of them. So we don't have this, in trying to make this figure, I'm not actually comparing a similar number of data points. So it's just said, okay, error, that's not going to work. So first I need to pull out the common set of plots. In this case, I use the function intersect, and I compare the first column from each of the matrices, which is the list of plot names. And so this is the set of common plot names between those two surveys. And I've made that an object called plots. this is just the note I mentioned about the column naming convention, and you might need to change that. So I've used that column name there, and that's the Linux default, but I'm pretty sure it's different on Mac, and it might be different on Windows as well. So I made this object called plots, and now I subset my matrices to only include the species that are in the common set of plots. So I've said, just using indexing, which species occur in this vector, which is the list of plot names, and so on and so forth for both the matrices I'm using. And now I try and plot them again, and I've added a trend line, and this is what I get. And if you look at it for a bit, you'll realize there's quite a big difference between the two. You're having almost 80 species in 66, but it's only a little over 70 in 2010 and the like. And so the slope of this relationship is not one-to-one, and it's not one-to-one even when you force it through the origin, even though this one isn't. Which suggests a decline in species number between these two time periods. Okay, so why such a big difference? Well, okay, step one is usually in Fenbosch surveys, we usually exclude what we call the seasonally apparent species, like geophytes, annuals, and we only analyze the permanently apparent species. So the seasonally apparent species are ones that you might only find them for a few weeks of the year. So if you happen to do your survey when that plant was flowering or had leaves, then you would count it in your survey. But if you did your survey 2 weeks later, you might not have found it. And because we can't come back and resurvey every plot every week for a year, the easiest way of dealing with this is literally just to exclude those species from your analysis and only look at the species that always have visible above ground biomass. And so what we do here is I exclude a set of species. And if we look at our spreadsheet again, one of the tabs is called excluded species. And if I read that in, that's a list of species names. So I'll cut those out of the analysis. I've also this includes aliens as well, because we were interested in indigenous biodiversity, not an alien biodiversity. So this excludes, or this tab of excluded species is a combination of seasonally apparent species and other undesirables. So again, we can exclude them using the same kind of subsetting approach as we did for matching the plots. And now we can plot that figure again. And it hasn't really changed much. So you're still seeing what appears to be a reduction in species numbers in 2010. So in this case, it's actually less than 70 now. It's less than 65 now, and it's almost 80 in 66. So there's quite a big drop there. But then I mentioned this issue of differences in post-fire age and differences in numbers of individuals. So ideally what we do is we actually, we can test if this is a problem. So there's two things you can do. The one is you can rarefy the data, which is you estimate the numbers of species you get for a given number of individuals sampled. And then the other thing is we can actually explore whether one sees a relationship between the change in species numbers and the change in vegetation age. So you would anticipate, that as a site gets older after a fire, there'll be a lot of species in the first few years because there's lots of individuals, and then that slowly declines through time. So if you have a site that in time 1 is young and in time 2 is old, you'd see a loss of diversity. Or an apparent loss of diversity. If you have a site that in time 1 is old and then had burnt before you survey it again, you might actually see a big spike in diversity. So we should work out if we need to account for that. And this allows me to bring in one of the other matrices, the environment data. So we have that environment spreadsheet that I mentioned, and we can pull that in. And this has a bunch of things like plot moisture class, plot number, the age of the vegetation in 1966, the age in 2010, so the age since fire, age in 66, and so on and so forth. classic issue number one when comparing multiple matrices is you might not have used the same labels or plot names. So in this case, these are just plots 1, 2, 3, 4, 8, 9, blah, blah, blah. And it doesn't have the CP underscore that I had in the species matrices. So you just have to paste them in, and that's very easily done with paste. So I've just taken, okay, rename this first column with, I realize I shouldn't have done that here. I should have renamed the plot column because, oh, no, it's fine. This works. Sorry, this works. So rename that first column by pasting together CP underscore and the number in the plot column with no separator, and you get the same plot labels. And then you can join the two. So I've used left join here, and it's joined by that ... one column. And essentially what that does is that the left join means take all the ones that you add all the data from end that match the ... column in species 10 and drop the rest. And it sorts them to match them as well. So you've aligned both data sets in one convenient function. You might be familiar with merge. It does pretty much the same thing, but it's usually much slower, and it sometimes does weird things. I find left join is a bit more stable. Okay, so now we can plot the change in species, in numbers of species against the change in vegetation age. And so here I create a matrix or a data frame, which is the change in species number, by just taking the row sums, the numbers of species in the 2 times and subtracting them, and then the age of the 2 times and subtracting them too. And you plot them against each other and add a trend line, and this is what you'd expect. So so this is the origin here at 0. If the plots got younger, they increase. If plots got older, they decrease in species number. If the plots got younger, they increase in species number. And so this is kind of a interesting interaction between fire and this relationship between abundance and diversity that one really has to take into account when analyzing Fembos data and a number of other systems, fire-prone systems in particular. Okay, so the other approach and what this suggests is that we really need to rarefy our samples. So I mentioned this a minute ago, and this is where you estimate the numbers of species you'd get for a set number of individuals. And how you do this is essentially you randomly sample sets of different numbers of individuals. So you say, okay, if I have 10 individuals, sample that 100 times or whatever, and on average, how many species do I get? Okay, then do 11 individuals, 12 individuals, 13 individuals, so on and so forth, and then you can get, that kind of describes your species accumulation curve. But if you want to compare a bunch of different sites, you would then, what you do is you take the lowest number of individuals across those sites, which in our case is 50, and you do this rarefying for just 50 individuals across all those sites, if that makes sense. So here we've said, okay, rarefy our species number, our species matrix by sampling 50 individuals for every plot. And it'll do it, I think by default it does it 100 times or something like that. I need to check the default. Then I've done the same for the 1966 and the 2010 plot, and then I plotted again. Okay, and this actually looks much better. So this is, I should have put them side by side here, but this is now species richness, not species number, against species richness. And the relationship is much tighter. And that's essentially because we're kind of dropping a bunch of this fire effect to some degree, although there are all kinds of interesting ecological reasons why we're not completely excluding the fire effect, and it's still worth investigating. But in short, we're much closer to comparing apples with apples here, and it's far more defensible. Interestingly, if you do a fit a linear model between the two and you force it through the origin, which you would expect to give you a slope of 1, it should be the one-to-one line. If you do that, you actually get a completely different slope. So you get a slope of around 0.8, and that does suggest that that there are lower species numbers in 2010. So if you look here, here there's 30 species, and the highest end here is about 24, I think, in 2010. So yeah, so it's showing a loss of species in these plots. If you want more information about why, you should read the paper. And we can also test this with a paired t-test. You can take these rarefied species richness vectors here, SR66 and SR10, and you can just do a paired t-test. So that essentially says, compare plot CP1 with plot CP1, compare plot CP2 with CP2, and so on. And that gives you a result that shows there's a significant difference between the two, and a kind of estimated decline in species richness of around 3.5 species, which is quite considerable. Okay, the last thing I'm going to talk about, and sorry if this has gone on a bit, it seems you guys have got pretty good staying power. We've only lost 2 people so far, but maybe some of you have put me on mute. It's always so weird talking to yourself. So the last thing to talk about is what we call the fourth corner, and this is, I'm just going to mention this because this is often the goal for a lot of community ecologists and a lot of analyses, which is essentially trying to establish the relationship between species traits and environmental variables. So you might want to know, how does leaf area change across a gradient of soil moisture or rainfall or that kind of thing. And this is called the fourth corner because it's usually not observed. So I mentioned earlier on, we typically have 3 data matrices: the kind of species by site matrix, the environmental variables by site matrix, and the traits by species matrix. But the fourth kind of important matrix that we can't actually measure in the field is the trait by environment matrix, and that trait by environment matrix we usually have to estimate, and there's a lot of different ways of doing it. There's multivariate methods, there's joint species distribution methods, there's all kinds of methods, and I'm just going to show you, I'm just kind of mentioning it here to make you aware of it. And the one example I show you here is essentially just kind of comparing the relative dominance of different functional types across different gradients. So for this, I pull in the traits spreadsheet, and this has the species, their families, and a bunch of traits like their growth form, herb, geophyte, graminoid, so on and so forth. And it's got this one called resprout post-fire, and that's essentially their post-fire regeneration strategy. Are the species killed by fire and have to recruit from seed, in which case they're an obligate seeder and they get a score, they're scored 0 in this column, or are they able to resprout from underground storage organs or epicomically from their branches and so on, and then they get a score of 1 and they're a sprouter. And so that's a big split in functional type in fynbos in general. And how to analyze that, this bit of code kind of extracts from this trait matrix, it extracts the list of species names that are sprouters, and then the list of species names that are seeders, which are the zeros. And then it's essentially just a vector of species names. And then what I'm going to do is I'm going to compare, what I want to do is I want to compare the relative, well, the richness of the species richness and the abundance of the seeders and the sprouters across a moisture gradient. So for in the environment matrix, there's a column called moisture class, and that all the plots were scored on a scale of 1 to 5, where 1 is a permanently wet site, 2 is seasonally wet, 3 is kind of seasonally moist, which is something that's usually kind of bordering a seasonal wetland, but it's not actually a wetland itself. And then 4 is kind of well drained, a site that doesn't really get additional moisture from drainage in the landscape. And then 5 is kind of like a well drained site, but it's got a north facing aspect or an equator facing aspect and shallow soils, which together kind of really make it more stressful for plants. So we have this 1 to 5 set of classes. Here I've kind of written some code that pulls together the different columns in a data frame. So this is a data frame. We want the strata, numbers of species for the stratas. And I've done this using the fancy tidyverse method I showed in the beginning. And I've done the same for the numbers of species of cedars, and then the abundance of stratas, the abundance of cedars, and then I've added the soil moisture class from the Envirometrics at the bottom. So that's just a table, which I should have shown you, but I haven't. But I'd like to turn it into a tidier table to help with plotting. And for this, again, I use the pivot longer function. And now I show off a little bit of fancy coding that you can use with pivot longer. So here I say, okay, take these 4 columns that I've created. So it's all of them apart from the soil moisture one. And remember, if you leave it out, it makes the soil moisture its own column. So that's here. And I say names to regeneration mode and variable. Before I only said names to one thing and values to another. Now I'm saying names to 2 different things and values to number. And you know, that's quite hard to get your head around, because how are you going to now put the values in 2 separate columns? And this pivot longer function actually provides all kinds of cool tricks for splitting up your column names in different ways. So here I've used a reg expression to say split the names into the before and after the underscore. And so here, these are 4 names, and they'll be split into strata and species, cedar and species, strata and abundance, and cedar and abundance. And then I put them into separate columns. So you've got a column for regeneration mode, which is essentially just strata and cedar, and a column for what I've called variable, which is species and abundance. You can come back and check this out and fiddle with it yourself if it's not making sense. But essentially, I've managed to split these columns and their names into completely different set of columns with strata and cedar, strata and cedar, and species and abundance, species, abundance. And then the number that's put in here are, you know, species number, abundance, abundance, species number, abundance, abundance, for the respective cedar or strata. Yeah, so an incredibly powerful function here, pivot longer and pivot wider, but it does take a bit of time to work out how to drive them without crashing them into a pole. So I'd recommend having a fiddle. And now we can plot them. And so this is what we got. So this is the 4, there wasn't actually any, I'd excluded the permanently wet sites from this data set. So there's only soil moisture classes 2 to 5, so running from wet to dry. And this is the abundances. And as you can see, there's really high abundances of cedar species at the wet sites, but that declines quite rapidly towards the dry sites. Whereas the resprouters show very little change. But funnily enough, in terms of species richness, the opposite is true. So both of them show an increase in numbers of species as you move towards drier sites. So it's a pretty interesting result. And there's a few things I haven't done or accounted for in this analysis. So before you read too much into it, give it some careful thought and think about things I've highlighted earlier that I haven't addressed in this little analysis. And I'm going to leave it there. Apologies, it was a bit longer than I was expecting, and I was hoping to be able to take more questions. But we have 3 minutes if you would like to ask any questions.